Alright, so I'm making this recording in one single shot. So basically, if I make any spelling mistakes around, or like I stutter or something, I apologize in advance. But time is of the issue because you have an exam tomorrow, so I'm just going to go, straight ahead and go through it. Alright, so first I'm just going to cover the epithelial tissues. So when we're looking at the epithelial tissues, we're going to start off with the simple ones, right? So we're going to do simple squamous epithelium first. And what you notice with the epithelial tissues is you're always going to have this free space, right? The free surface, because of the fact that these epithelial tissues, they line body cavities, okay? So you're going to notice when we're looking at different epithelial tissues, there's open spaces. And this is not the best example, mostly this one right here and over here. But for example, we're going to see that as characteristic of epithelial tissue. But for simple squamous, we're only looking where the arrows are shown right here. So when we're looking at the arrows, I see that these nuclei look all very flat and their cells are very flat. And remember, squamous means flat. Okay? So when we're looking at this, we're looking at one layer of just the simple squamous epithelium lining this this body cavity here. Okay? So remember, they're not going to ask you for this area right here. Okay? They're just going to ask you at the free surface right there. This is just another example in the lungs right here. So in the lungs, where these are shown are the different nuclei. So probably the best examples are right here and right there. But you see all these different little flat nuclei lining these air pockets, if you want to call them, alveoli, they're supposed to be called, but that's A and P too. But these air pockets within the lungs. So simple squamous are just basically lining these areas, making these little pockets, okay? So this is one simple squamous cell, this is another simple squamous cell, etc. But remember, this is just one layer, okay? This is another example. Remember, we're just looking at the arrows, where we just have this one nucleus right here, and it's a one cell. Here's another example, one nucleus, one cell, and you can see that happening all across here, just this one little line. They might not show you this on the exam. I'm only putting this on the just in case that they put a pointer right there. Simple squamous can possibly be asked in this area within the kidneys, okay? But the best examples are really this one and really these ones that show the free surface in the very flat cells. Okay, I wanted to jump to stratified squamous right after. Remember when I said about stratified, the part that's going to look like the actual cells, like the flat nuclei, are going to be at the top, at the apical layer. And at the basal layer, they're never going to really look like that because those are the areas that are constantly regenerating. But just know that there's multiple layers here. And this is stratified squamous. And I believe for stratified, the only one they're going to ask you is stratified squamous. I don't think they're going to ask you stratified cuboidal or columnar because it becomes more difficult to identify them when you're basically comparing them to, let's say, transitional or pseudostratified ciliated columnar. So remember, when you're looking at stratified, it's probably just going to be stratified squamous. Okay, so you see the flat cells at the top over here, and this is just multiple layers of just the squamous. So this is stratified squamous. Here's another example where you have the basal layer and then you have these layers going up, and they don't look squamous at the bottom, but as you start to get to the top, they start to flatten out. So this is another stratified squamous. Same exact thing, don't really have to go over this one, and basically same exact thing here. After this, I wanted to jump to actually to skin, because skin actually does have stratified squamous all the way in the epidermal layer, right? Because you have the epidermis and then you have something called the dermis, but the whole thing is skin. But when they show you skin, I believe they just want you to say it's just skin. So I wanted to put skin next to stratified squamous to show you basically I think the differences on how they're going to show it. I believe skin is going to be more zoomed out, and when you're looking at skin, you notice these very nice color progressions between the layers of the epidermis. So I see this nice little purple, it gets a little darker, then lighter, then very much lighter, then a little bit darker here. And you kind of see this as characteristic when you're looking at skin. Here's another example. We're just starting off from right here, and we see these nice little layers of the stratified squamous as we move along to the top. This is actually a good example because we're starting just here and we see all these different layers, but you can see all the dying off cells here, which is characteristic of skin, because remember, you always have skin dying off at the apical surface. It's always regenerating. But remember, for skin, you just have to say it's just skin. Okay? When you see something like this, you're going to say it's stratified squamous. They're going to be more zoomed in. This is a more zoomed in picture. And you can see that they're not going to be really incorporating multiple layers all the way to the bottom. Right? This, you can see the difference between this one, which is stratified squamous, and it's very light here, and it's not too many layers of different colors. And then you look at this one, where these layers are definitely separated between the colors, between the staining dyes and stuff like that. So remember, just say it's skin when you see that little nice progressive color change from top to bottom. And then for stratified squamous, they're going to be more zoomed in, okay? And they're just going to show at the apical layer they're flat cells, and it's just multiple layers. Alright, so now we can move on to the simple cuboidal, and this is actually a very good slide here. It shows the cubic cells here, and remember, the nuclei are always going to look like the shape of the cell. So when the squamous ones were flat, the cuboidal epithelium will have nice little circlish, nuclei. Basically, it's going to look like someone like a cube. In this case, it's obviously circular, but they're going to basically be relatively the same shape as their cell. So this is simple cuboidal, and simple cuboidal tends to be in a circular shape here. They tend to make these little circles, and here's another circle. I want to add another one right here. It's basically the same exact thing. Forms the little circles. You see the nice little cuboidal shapes and the nice circular nuclei. You're going to have slides where they're very well stained versus cells that are very, not as well stained. So I wanted to make sure I had both kinds of stains. This one, you see the circular nuclei in the cuboidal shaped cells in the circular fashion. Same exact thing. And then I just wanted to show this last one because, yes, they're usually in this circular fashion, but they do also sometimes go in a straight line like this. But you can see that it's just the circular nuclei and the circular cube-shaped cells for simple cuboidal. Next, we can move on to simple columnar. And I apologize if I'm going too fast, but if this video is too long, it's going to take way too much of your time. But for simple columnar, you're going to notice that they're going to have these nice nuclei. They're more so longer, kind of like their cells, versus the circular nuclei of, let's say, the cuboidal. So this is actually a very good example. This one's a little bit taller, kind of like the actual cell. So this is simple columnar. They're column shaped, and then here you also see that little free surface that's characteristic of epithelial tissue, because remember, epithelial tissue, they line cavities, so you're always going to see a free surface. Okay, you're going to see the basement surface, the basal layer, and then the apical layer, which is where you're going to see the free surface. So this is simple columnar, and this is actually non-ciliated. You see just smooth tops here at the apical layer. This is another example. You see the free surface. All these nuclei tend to always run in the same level when you're looking at simple columnar. That's going to be a good way to differentiate between this one and the next one we're going to cover, which is this one right here. The nuclei do not run in the same level. They're all over the place. But we'll get to this in a little bit. I just wanted to show how in simple columnar, all the nuclei are in the same level, right? A little bit taller than they are circular. And then obviously you see that they line certain areas, so you see the free open space. This is another example of simple columnar. This one also, there's your free space, and you can see the nice nuclei on the same level in the column-shaped cells. We already did this one. And I wanted to show this one because this one is more zoomed out versus these ones. These are a lot more zoomed in, but they might have more zoomed out pictures. More zoomed out slides when they're looking at the microscope. So this one is a little bit more zoomed out, but it's the same thing. You can see these very tall-like nuclei, and then how they are uniformly in the same level. All these nuclei lined up very nicely. Alright, so now we can move on to the one that may confuse others, differentiating between if it's pseudostratified or if it's just a simple columnar. But you can see the same level nuclei in simple columnar. But for pseudostratified, you see that the nuclei are top to bottom, top to bottom, top to bottom in that orientation. And the thing is though, you may look at this and you might say, okay, I see multiple layers here, but that's not the case. It's just that these cells are so compact that the nuclei are basically either pushing upwards or they're pushing downwards. And that's why they call it pseudo stratified. Pseudo actually means false, so it's basically false stratified columnar epithelium. But the main thing that's important about looking pseudostratified columnar epithelium is, actually I forgot to write it here, but it's obviously pseudostratified ciliated columnar epithelium. You see all these beautiful cilia here. The main goal of these cilia is just to move along mucus along, let's say, the respiratory tract. So that's why they have these cilia here. So the main thing you want to look at for pseudostratified is looking at these nuclei with their top to bottom, and they're always going to have that cilia. You're going to see also these little white cells here. It's a lot better seen on this slide. But basically these little cells, you don't have to know what they are, but they just produce mucus. But you're going to see them just around when you're looking at pseudostratified columnar epithelium. And it's very good when you're looking at the nuclei here to compare that to this, for example. Very different between simple columnar and pseudostratified columnar. And keep in mind also you have the free surface there, very characteristic of epithelial tissue. Same thing over here. And this actually has very good cilia when you're looking at this one. And then this one actually has very good cilia also. So that's pseudostratified columnar epithelium. And I wanted to keep this one close to the next one we're doing, which is transitional, because people tend to mix this one up because of the nuclei. Kind of looks similar to these ones. But the most important thing is the apical surface, okay? When looking at the apical surface, the free surface area for pseudostratified, you see all the cilia. It is not smooth, okay? Let's compare that to this right here, which is transitional epithelium. There's absolutely no cilia, and those are very smooth surfaces at the top, at the apical layer. So that's the most important when differentiating between transitional and pseudostratified, is look at the surface, okay? If it's smooth and it has all these nuclei all over the place, it's going to be transitional. If it has the cilia coming out, it's going to be the pseudostratified columnar. So this is just one side of the transitional epithelium, and this is really the same exact thing. You see the nuclei running all to the top, and very smooth surfaces here. And that's basically the same thing as the last slide. But basically they call it transitional epithelium because these cells, they transition between columnar shaped or cuboidal shaped. For example, like in the urinary bladder, if the urinary bladder is distended, has enough urine, it's going to want to basically stretch, right? But if the urine is voided from the urinary bladder, then it's going to want to basically shrink. So that's why they call it transitional epithelium, because these are areas within the body that either stretch or shrink depending on how much fluid is in there, and they transition between a certain shape and another. This is just another example here, looking at, you see once again epithelial tissues have three surfaces, flat tops, and no cilia at all. Okay, so this is transitional. This one's going to show a little bit more how it closes off. The best way to show it is actually in this one. This is more of a zoomed out example of transitional, but it's no different. You can see the nuclei running to the top, the very smooth surface, and actually this is not the best example. This one is way better because this shows clearly a smoother surface. These tend to get a little damaged when fixing the slides, so this is not the best example. This is way better example where you see the smooth surface. Just remember that this is a very zoomed out picture. We're really looking at this area right here when we're looking at, for example, this one, or this one. But remember, when it zoomed out, they formed these closed surfaces. For example, in the urinary bladder, I believe this would actually be a ureter, but here would be the urine in here. And this has no urine, therefore all the cells are basically shrunken. So that's transitional epithelium. We finished the epithelial tissues. This is the more easier part, the connective tissues. Let's do loose areolar connective tissue first, because this is definitely the easiest. You're going to see all these fibers, and actually that's what's important about connective tissue, is it's always full of fibers. Okay, so you see all these little fibers moving along across, and there's these very little pink ones in the back too, not as darkly stained, but that's the point. They're different types of fibers, therefore some stain lighter, some stain darker. But you're going to see all these, it almost looks like a webbing. I think some people say it looks like, actually no, forget about that. But it just does look like just mostly webbing. And you do see many empty little spaces here. That's why they call it loose connective tissue, because it's not dense. It's not densely packed cells. If we compare that to, let's say, these are incredibly densely packed fibers. Okay, versus, and this would be dense connective tissue versus loose, where you see many open spaces. But keep in mind, connective tissue, they don't have free surfaces like epithelial. Okay, that's a way you can basically differentiate between the two. The connective tissues do not have free surfaces. They do not line cavities like epithelial tissue do. Okay, and this is the one we looked at, but for the other two examples I have for loose areolar connective tissue are the same thing. It looks like, webbing, and you see little empty pockets everywhere. Same exact thing here. So that's loose areolar connective tissue. Adipose connective tissue, you see each of these are one little adipocyte, one little cell of fat. And the main distinct thing looking at here is where they have pointed at these little pointers, is that there's so much fat inside the cell that actually pushes the nucleus outwards. So you're always going to see the nuclei for the adipose tissues always on the periphery. These are all good examples. So this is adipose tissue. It may look like it's open pockets, but really these are actually cells themselves with fat within them. This is another example here, the peripheral nuclei, which are pushed to the outside because of the fat within them. And some people would get confused looking at this and saying, okay, what's the difference between this and simple squamous then? Well, I put these two slides together with adipose in the simple squamous found in the lungs that I showed you earlier. You can tell when looking at these cells, which this is just one cell right here, one cell, versus this is one little cell, this is one little cell, and this is one little cell. This is all a free open surface right here. This is all free surface. This is just actual spot with fat in it. But the most important thing when looking at the orientation of everything, you see nucleus after nucleus after nucleus after nucleus over and over in simple squamous. Okay, you do not see that in adipose. In adipose, if you're comparing these two, you would expect a nucleus, a nucleus, a nucleus, just like that. But these cells are much bigger, and the nuclei are just going to be basically in, I don't know what's the word I'm looking for, in further areas separated. For example, from here you don't see a nucleus for a while until you get to about here. Okay, because these are just one cell and one nucleus, one cell and one nucleus, and that it just tends to be like that. So you can tell the difference also. These are more smoother droplets of fat, creating a more pushed out smoother surface, versus this is all zigzaggy. So this would be simple squamous, all open space. This would be adipose, one cell and just one nucleus per the whole cell. So just looking at these two, try to see the difference yourself. Okay. Lastly, we're actually almost done here, where the dense regular connective tissue, you see the fibers running very smoothly across. And don't worry about this or this. We're mainly looking from here to here. Okay, but you notice that all these nuclei are very flattened because the fact that every fiber is so densely packed that they're squeezing the nuclei, but the nuclei are just running almost like kind of like a school of fish, if anything. But the fibers, and it could look very smooth when looking at dense regular connective tissue. But I wanted to put in slides such as this one, where the staining is not always very smooth, but you still see the nuclei going in a straight line, almost like a school of fish swimming, and the fibers in between just running straight. They tend to make this little curvy orientation, but really it's just going from one side to the end, so it's basically straight. This is another example. You see all the nuclei running like a school of fish, flattened because of how densely packed the fibers are. So this is another example of dense connective tissue. And all these slides I'm going to put into a Quizlet for you, so you can basically just practice with the same exact slides I gave you, and hopefully by using the Quizlet and the little thing that they have, I think it's called scatter or just test, you can do mixing matching and kind of get this more memorized in a quickly possible. Watching this video maybe once might be enough. Maybe watching it twice might do it. Remember, don't watch it more than twice because this video is going to be pretty long. So just stick to really the Quizlet after this to really, for time management purposes. So hopefully that worked out well for you, and hopefully this helps.